Many human actions are reflexes. Which two of the following are examples of reflex actions? Tick two boxes. Well, reflex actions are responses to stimuli that are unconscious and instantaneous, or rapid. Now, which of these fit that description? Let's go through them. Jumping in the air to catch a ball. Choosing to catch a ball is a conscious choice, so this is not a reflex action. Next, we have raising a hand to protect the eyes in bright light. This itself is not a reflex action, although it's easy to get mistaken, because there are reflex actions which involve responses to bright light. However, these occur within the eye. The next is releasing saliva when food enters the mouth. Now this is a reflex action. The stimulus is the entrance of food into the mouth, and the release of saliva is unconscious. One cannot choose to release saliva. Instead, it happens automatically as a response to food. Next, running away from danger. And although this is a response to a stimulus, this requires the brain, and it's a conscious decision. Finally, withdrawing the hand from a sharp object. This too is also a reflex action. The sharp object is a stimulus which results in a rapid movement of the muscles of the hand without requiring the use of the brain. So these are the two answers. Figure 1 shows how the size of the pupil of the human eye can change by a reflex action. Name one stimulus that would cause the pupil to change in size from A to B, as shown in Figure 1. Well, looking at Figure 1, we can see that the pupil, the black circle in the center, has become smaller. This means less light will be let into the retina, because the pupil is the hole through which light enters the eye. Well, what stimulus would mean that the eye needs to let in less light? Well, this would be bright light. Since if the pupil remained the same size, the retina may be damaged by the bright light. Structure Q causes the change in the size of the pupil. Name structure Q. Let's have a look back at Figure 1. Well, Q is what surrounds the pupil, and this is known as the iris. And this is what gives people the eye colour that you see. Thus, the iris is our answer. Describe how structure Q causes the change in the size of the pupil from A to B. Bear in mind this is a 1-mark question, so we need only 1 point. Let's draw a simplified version of the iris and pupil. When changing from A to B, the circular muscles, which were relaxed, contract. They therefore become thicker, reducing the size of the pupil. We do not need to go into this much detail for our answer, and can state muscle contraction as the cause. Figure 2 shows some structures involved in the coordination of a reflex action. Describe how the structures shown in Figure 2 help to coordinate a reflex action. Now, it's a 6-mark question, which is level marked. And to get the full 6 marks, we need to make our answers detailed and coherent. Well, let's go through Figure 2 and begin by labelling the neurons. Neuron A is the sensory neuron. Neuron B is the relay neuron. And finally, neuron C is the motor neuron. The process of a reflex action begins with a stimulus, which is detected by the receptor. Then, an impulse is created, which travels through the sensory neuron. This travels all the way to the spinal cord. At the spinal cord, there is a synapse. Now, a synapse is the gap between two neurons. Within a neuron, the signal travels by electricity, an electrical impulse. However, between neurons, chemicals, known as neurotransmitters, are released. They diffuse through the synapse and are taken up by the adjacent neuron, in this case the relay neuron. The uptake of this neurotransmitter results in an impulse being sent through the next neuron, in this case the relay neuron. The impulse is conducted through the relay neuron before passing to another synapse between the relay neuron and the motor neuron. Following this, it moves through the motor neuron to the target organ, and the effect occurs. This is generally a muscle or a gland, and will result in either the contraction of a muscle or the release of chemicals from a gland. Let's write this information into our answer. First of all, a receptor detects a stimulus. Now for extra detail here, we're going to give an example, such as heat. This generates an impulse which is conducted through a neuron. Conducted is the same as saying that it travels through said neuron. The neuron in question is neuron A, a sensory neuron. The impulse travels to the spinal cord, where it reaches a synapse. Neurotransmitters are released, which are absorbed by B, the relay neuron, stimulating an impulse through it. The signal moves through another synapse into the motor neuron, C. The impulse moves through this to the muscle, stimulating contractions. Now instead of stating muscle here, you can instead have mentioned the gland and stated that it stimulates the release of a chemical. Although this question is level marked, we can look roughly at where the marks will be awarded. The first, for discussing the detection of a stimulus by a receptor. The second, for talking about the conduction of an impulse. Next, for talking about the movement of this impulse to the spinal cord. Next, for stating and describing what happens in a synapse. Then, for the naming of the neurons correctly throughout the answer. And finally, for discussing the effect of the reflex, that is, the muscle contraction. Many scientists think that global air temperature is related to the concentration of carbon dioxide in the atmosphere. Figure 3 shows changes in global air temperature and changes in the concentration of carbon dioxide in the atmosphere. Looking at Figure 3, you can see the time along the x-axis go from 1960 to 2010. You can also see the change in air temperature on the left y-axis and the concentration of carbon dioxide in the atmosphere on the right y-axis. The question asks us to complete Table 1 and to use information from Figure 3 to do so. Choose answers from the box. You may use each answer once, more than once, or not at all. The options are constant, decreasing, and increasing. Looking at the table, we need to discover the trend between 1960 and 77, between 1977 and 2003, and finally 2003 to 2015. Let's have a look at the figure to complete the table. Well, first of all, looking at air temperature, let's first isolate the dates that we need to look between. First of all, up to 1977, then between 1977 and 2003, and then 2003 onwards. Drawing a line between these dates makes the trend more easy to see. First of all, we can see a downward trend to 1977, followed by an upward trend, and then a stabilizing of the trend from 2003 onwards. On the other hand, with carbon dioxide, there is an increase for all three periods of time. Let's fill in these things on the table. Well, first of all, for carbon dioxide, all three boxes will have increasing, and for air temperature, it'll be decreasing, followed by increasing, and finally constant. You get 1 mark each for each of the rows. Many scientists think that an increase in carbon dioxide concentration in the atmosphere causes an increase in air temperature. How would an increase in the concentration of carbon dioxide in the atmosphere cause an increase in air temperature? Well, carbon dioxide is a greenhouse gas. This means that it allows shortwave radiation from the sun to pass through. However, the longwave radiation that is emitted by the Earth is reflected back. Thus, it acts like a greenhouse, increasing the temperature of the atmosphere, since less heat is lost. For 1 mark, we need only 1 point, so we'll say traps heat. Alternatively, you could say insulates or reduces heat loss. Evaluate evidence for and against the theory that an increase in the concentration of carbon dioxide in the atmosphere causes an increase in air temperature. Use data from Figure 3 and your own knowledge. This is a 4-mark question which is level marked. And for level 2, that is 3 or 4 marked, your answer needs to be detailed and coherent. Since we need to evaluate the evidence for and against, we must ensure that we discuss both sides of the argument. Let's have a look back. It is important to maintain water balance in the body. Figure 4 shows how much water a person gained and lost by different methods in one day. Let's have a look at Figure 4. Well, on the left we can see one bar graph with the water gained by the body. Now, this is by three means: food, drink, and metabolism. Metabolism includes processes such as respiration, which produce water. On the right, we can see the water lost from the body. Now, this has all but the feces filled in, which includes the urine, skin, and breathing. As we can see, the majority of water is lost through the urine. Let's see what the question wants us to do. When water is balanced, the volume of water taken into the body is equal to the volume of water lost by the body. Calculate the volume of water that the person lost in one day in feces. Use information from Figure 4. Looking at Figure 4, we know that since water is balanced, the total water gained by the body is equal to the total water lost from the body. Therefore, let's calculate the total water gained first of all. The amount of water gained in the three different categories are 500, 1,500 and 400 respectively. Therefore, in total, the water gained is 2,400 centimeters cubed. In Figure 4, we can calculate the total water lost excluding feces. This is the same as 1,500 plus 400 plus 380, which is the same as 200, 280. And to find feces, we find the difference between these two values. 2,400 minus 2,280 is 120. This is the volume of water lost in feces. Here, one mark comes from the working, and the second comes from the correct answer. Figure 4 shows that one method of gaining water is by metabolism. Which metabolic process produces water? Tick one box. We have the options of the breakdown of protein to amino acid, changing glycogen to glucose, the digestion of fat, and the respiration of glucose. Respiration is the only one of these which does in fact produce water. The equation for respiration is glucose plus oxygen is converted into CO2 and water. Here, water is produced. The other three processes actually use water in the breakdown of the said molecules. Therefore, the respiration of glucose is our answer. The next day, the person ran a 10-kilometer race. The volume of water lost from the body through the skin and by breathing increased. Explain why more water was lost through the skin during the race. Well, water is lost from the skin as sweat, produced in sweat glands. The reason this is done is to cool the body. As the sweat evaporates, this requires energy. This takes heat energy away from the skin and the body, thus cooling the body. This is important during exercise, since exercise increases the temperature of the body. Exercise, running in particular, creates lots of heat because the muscle contraction required needs more aerobic respiration, which is a reaction that produces heat. So let's write our answer. We're first of all going to say more sweating, and we're going to follow this with the reason why, to cool the body. Instead of saying to cool the body, you could alternatively have said because running produces heat, or to regulate the temperature of the body. Explain why more water was lost by breathing during the race. Let's think about what occurs when one runs. Well, when you're running, the muscles must contract, and this requires lots of energy. So more energy is needed. This energy comes from respiration, therefore a greater rate of respiration is required. With more respiration, more oxygen is needed, since it's a reactant of the process. Therefore, we need to have a greater breathing rate so that more oxygen can be taken up by the body. Water is contained in the air that you breathe out. Therefore, with a greater breathing rate, meaning that a greater volume of air is breathed out, more water will be lost. thus answering the question. During the race, more energy was needed for muscle contraction. Therefore, more aerobic respiration is required, since this is the process that produces energy for muscle contraction. This uses oxygen, so a faster breathing rate was necessary to take up more O2. The marks here come first of all from talking about more energy being needed, then for saying that aerobic respiration is required for this energy, and finally for saying that a faster breath rate is required to supply more O2. Some students investigated the size of a population of dandelions plants in a field. Figure 5 shows the field. Notice the woodland to the south of the field, which will reduce the light intensity on the area of grass to the south of the field, which could affect the dandelions. The students placed a 1 meter by 1 meter square quadrate at 10 random positions in the field. Counted the number of dandelion plants in each quadrat. Table 2 shows the students' results. We can see the 10 quadrats and the number of dandelion plants, which ranges massively. The question asks us, why did the students place the quadrats at random positions? Well, the reason for using random positions is to make the study representative of the entire field. You could also have said that it's to reduce bias. So if you chose yourself, you could subconsciously choose the areas with the most dandelions, which would skew your results. Or you could also have said because there was an uneven dandelion distribution in the field. Estimate the total number of dandelion plants in the field. Calculate your answer using information from Figure 5 and Table 2. Give your answer in standard form. There's a 5-mark question, so we must include our workings to get the full marks. Let's have a look at Figure 5 and Table 2. Well, each quadrat is 1 meter by 1 meter. There is a 1 meter squared area. So, the process we'll undertake is we'll find the mean number of dandelions per meter squared by calculating the number of dandelion plants on average per quadrat. We will then find the total area of the field. Now, since we know the number of dandelions per meter squared, we can multiply this by the total area of the field in meter squared to get an estimate for the total number of dandelions. We must then convert this to a standard form. So first of all, to find the mean, we add up each of the data points for the number of dandelions, and then we divide this by 10, since there are 10 different quadrats. This gives a value of 6.0, or 6. Next, let's find the area of the field. Since this is a compound shape, we'll split it into 2 rectangles. Let's first find any unknown lengths. The length here is 200, since it is 300 minus 100. The area, therefore, in this rectangle is 150 times 200, which gives us a value of 30,000. In the bottom rectangle, the value we don't know for the length is 250 meters, 150 plus 100. Therefore, the area is 250 times 100, which gives us a value of 25,000. Adding these together, we get a total area of 55,000. We now need to write this into our answer. The key figures to mention are first of all the mean that we calculated, followed by the area, and then we need to calculate an estimate for the number of dandelions. So let's first of all state the mean dandelions per meter squared, which is 6. The total area is 55,000, and as we already stated, the total dandelions will be the product of these two, 6 times 55,000, which gives us a value of 330,000. This is not in standard form, and to put it into standard form, we have to have a number between 1 and 10 multiplied by 10 to the power of something. 3.3 will be our base number, and to find the power to which 10 is raised, we're going to count the number of digits to the decimal point after the first 3. This gives us a value of 5, so it's 3.3 times 10 to the 5. This is our final answer. Quadrats 5, 7 and 8 were each placed less than 10 metres from the woodland. These quadrats contained low numbers of dandelion plants. The student made the hypothesis. Light intensity affects the number of dandelion plants that grow in an area. Plan an investigation to test this hypothesis. This question is level marked, and to get the top band of 5 or 6 marks, we will need to produce a valid outcome from the method that we describe. We also need to have clear steps with a logical order. So let's think about how we're going to carry out this experiment. Well, we're comparing light and dark, so we want to have 2 different areas, one that is light and one that is dark. And we're going to treat each the same way. The way we'll treat them is by taking samples, by using quadrats and placing them at many locations on each. When we place the quadrats, we will then count the number of dandelions present in each, as well as recording the light intensity. The method of choosing the location of these quadrats is very important. As the question previously mentioned, it's vital that they're chosen randomly to avoid bias. To do this, we'll divide the areas into a coordinate grid. Then, using a random number generator function on a calculator, we'll choose the set amount of coordinates at each location. Let's write our answer. First of all, identify 2 areas, one that is light and the other that is dark. At each, divide the area into coordinates. This makes the choice of quadrat location quantitative, so we can use a random number generator function. If one were to go into the field and simply choose locations, there is a large degree of subjectivity, which would undoubtedly cause bias. Use a random number generator function on your calculator to generate 20 coordinates for each place. 20 is a large number, and using a large number ensures that our samples will be representative of each of the fields we are studying. At each of these, place a 1 meter squared quadrat and count the number of dandelions. Measure also the light intensity at each. Record these. Compare the data at the different light intensities. Now, although this is level marked, let's go through our answer and see where the marks could come from. First of all, from identifying a light and dark areas to study. Next, from using coordinates. Then using a random number generator function. And also using a large number of coordinates. The placing of the quadrat and counting of the dandelions is another. And also measuring the light intensity. That's 6. However, I'm sure you could also get another one for comparing the data at the different intensities, were you to forget any of the other ones. Cell division is needed for growth and for reproduction. Table 3 contains 3 statements about cell division. Complete Table 3. Tick 1 box for each statement. Let's have a look at the statements first of all. The first one says that all cells produced are genetically identical. The second says that in humans, at the end of cell division, each cell contains 23 chromosomes. And the third is that it involves DNA replication. And we're asked to state whether these occur in mitosis only, meiosis only, or both mitosis and meiosis. Let's think about the process of mitosis. This produces two genetically identical daughter cells. On the other hand, meiosis produces four genetically distinct daughter cells. Therefore, the first statement is true for mitosis only. Let's think about the second statement. Well, normal human body cells, also known as somatic cells, have 46 chromosomes. That is 23 pairs of chromosomes. Within each pair, one chromosome comes from each parent. In mitosis, 46 chromosomes are also present in all daughter cells. However, during mitosis, which produces gametes, they only have 23 chromosomes. So, the second statement is true of meiosis only. DNA replication occurs in both mitosis and meiosis, hence is true for both. Bluebell plants grow in woodlands in the UK. Bluebells can reproduce sexually by producing seeds. Bluebells can also reproduce asexually by making new bulbs. One advantage of asexual reproduction for bluebells is that only one parent is needed. So just two other advantages of asexual reproduction for bluebells. Well, first of all, we can say that many offspring are produced. This is due to the relative ease of asexual reproduction compared to sexual reproduction. A second advantage is that it takes less time, as there is no need for pollination or for the fertilization of gametes, all of which takes a long time out of the process of reproduction. Alternatively, we could also have said that the process is more energy efficient. Or, alternatively, that it allows colonization of a local area, since many individuals will be produced within a small area, colonizing it. Other correct statements would also receive a mark. Explain why sexual reproduction is an advantage for bluebells. Let's think about sexual reproduction. Well, sexual reproduction requires gametes. These are produced by meiosis. The process of meiosis produces genetically distinct cells. Therefore, genetic variation is introduced. Hence, some individuals will be better adapted to survive in the conditions present in the environment. These will be our first 2 points. The sexual reproduction results in genetic variation in an offspring. So, some individuals will be better adapted to survive. For plants in particular, sexual reproduction generally uses seeds. These seeds can travel large distances before growing into plants. Thus, this allows the plants to colonize new areas. On the other hand, asexual reproduction creates new genetically identical plants in the neighboring vicinity to the original plant. Therefore, this dispersal of seeds allows the plant to spread to other areas. So let's write this down. Seeds may disperse long distances, allowing colonization of new areas. Finally, we could also say that many offspring are produced in this process, so there's a higher probability that some will survive. Looking at where the marks come from, the first and second are from talking about the introduction of genetic variation, and this making the plants better adapted to survive. The third comes from discussing the colonization of new areas. And finally, the fourth mark from discussing the fact that more offspring creates a higher probability that some will survive. Some students investigated geotropism in the roots of bean seedlings. Figure 6 shows the apparatus used. Let's have a look at Figure 6. The main difference here is that apparatus A is stationary and apparatus B is rotating slowly, using a motor. Both have a cork mat onto which beans have been pinned. From the beans, roots are growing, which will presumably be measured. Let's read the method. This is the method used. 1. Measure the length of the root of each of 10 bean seedlings. 2. Pin 5 seedlings to the cork mat in apparatus A. 3. Pin 5 seedlings to the cork mat in apparatus B. 4. Leave A and B in a dark cupboard for 2 days. 5. After the 2 days, make a drawing to show the appearance of each seedling, and also measure the length of the root of each seedling. The first part of the question asks, why did the student surround the seedlings with damp blotting paper? Tick one box. The options are to prevent light affecting the direction of root growth, to prevent photosynthesis taking place in the roots, to prevent the growth of molds on the roots, or to prevent water affecting the direction of root growth. Well, the damp blotting paper provides water to the seedlings, so this is what it's concerning. Thus, the first two of the statements are incorrect, since they concern light. The presence of water would in fact encourage the growth of mold, so this is incorrect. Therefore, the final statement is correct, to prevent water affecting the direction of root growth. This is useful since the direction of root growth is what we are studying. Apparatus B is a control. Apparatus B rotates slowly. How does apparatus B act as a control? Well, we're thinking about the effect of gravity on the direction and size of root growth. So let's think about the effect of gravity on the seedlings when it rotates. Well, as the drum rotates, all sides of the bean experience the same amount of gravity. So, if we picture the bean, over time, gravity acts evenly on all sides. So this is how B acts as a control, so that we can see the effect of gravity acting from one side only on root growth. Table 4 shows the students' results. Let's have a look at Table 4. We have both apparatus A and B, and for each, the seedling number, length at start, length after 2 days, length change, and mean length change are shown. We can see that for apparatus A, there appears to be a higher change in the length of the roots. One student stated the mean length change for the seedlings in apparatus B is not valid. Suggest the reason for the student's statement. Well, looking at the results for apparatus B, we can see that for seedling 4, there is only a change of 1 mm. This is clearly an anomaly, since the other ones range between 12 and 15. Now this anomaly has been included in the calculation of the mean. By eye, you can tell this, since the mean is lower than any of the other values for the change. If it were excluded, the mean would in fact be 13.75. Our answer here then is that the anomaly of seedling 4 has been included when calculating the mean. Suggest one improvement the students could make to obtain a more valid mean length change for the seedling in apparatus B. Well, as I've just done, first thing you could do is to recalculate the mean without the anomaly. That is, using seedlings 1, 2, 3, and 5 only. Alternatively, you could repeat the experiment and then recalculate the mean. Figure 7 shows the students' drawings of the 2 seedlings at the end of the 2 days. You can see on the left, seedling from apparatus A, which has a curved downward root. On the other hand, the seedling from apparatus B has a straight root. A plant hormone is made in the root tip. The hormone diffuses from the tip into the tissues of the root. Explain how the hormone causes the appearance of the seedlings in figure 7 to be different. You should refer to both seedlings in your answer. The question asks us to explain. So we need to both state what is happening and how this causes the difference. So, let's have a think about what's happening. If we imagine a root tip here with the upper and lower cells shown, gravity acts downwards, so the auxin produced in the root tip is present in greater quantities at the bottom of the root tip than the top. The auxin reduces the cell elongation. Thus, cells at the top elongate greatly more than those at the bottom. This causes the root tip to bend due to the uneven rate of growth of cells. Let's write our answer using this information. There is more auxin at the bottom of the root in A, but even distribution throughout the root in B. Therefore, there is reduced cell elongation at the bottom cells of the root in A. This process causes the root to bend. Instead of discussing root cell elongation here, you could also talk about the rate of growth. For the last point, you can instead have said that the top grows faster than the bottom. The 3 marks are gained here by talking about the uneven distribution of auxin in A, the even distribution in B, and the result of this in A. That is, that there is a difference in the rate of cell elongation or cell growth rate at the top and bottom of the root. In horticulture, plant hormones are used for controlling plant growth. Draw one line from each plant hormone to the correct use of that hormone. The 3 hormones here are auxin, ethine, and gibberellin. As we've just discussed, auxin is involved in the growth of roots. Therefore, it's used in the industry to stimulate root growth in plant cuttings. Ethine is a plant hormone which ripens fruit. Therefore, it can be used to reduce the time taken for tomatoes to ripen. Finally, gibberellin promotes seed germination. These are our answers. For some extra information, ethine is actually produced by bananas. This is why, when you perhaps put bananas next to other fruit in the fruit bowl, they cause other fruit to ripen. Figure 8 shows a food chain for organisms in a river and the biomass of the organisms at each trophic level. We can see the biomass is given in grams per meter squared. The food chain includes a producer, the algae, a primary consumer, the invertebrate animals, a secondary consumer, small fish, and finally a tertiary consumer, large fish. Draw a pyramid of biomass for the food chain in figure 8 on figure 9. You should use a suitable scale, label the x-axis, and label each trophic level. Let's have a look at the grid given in figure 9. For ease, I'll write down the biomass of each trophic level here. We want our pyramid to fill as much of the figure as possible. We're creating a pyramid, so half of the biomass will lie either side of the pyramid. Therefore, on either side of the 0, the scale must go up to at least 840 divided by 2, which is 420. Therefore, each of the larger squares will be worth 50, so that it goes up to 450 in total. So, let's label the squares accordingly. Remembering that the left-hand side is a reflection of that to the right-hand side of the central vertical line. Be careful not to add negative values to the left-hand side of the 0 out of habit. To work out where to plot either side of each section of the pyramid, we're going to divide each value of biomass by 2. We can then plot these bars to form a pyramid. starting with the largest, that is the producer, the algae. Be careful here that you plot it accurately, since you are only given or plus or minus half a small square of leniency. Once we have plotted the pyramid, we must then make sure to label it. First of all, we'll label the x-axis as biomass and include the units, grams per meter squared. Then, we have to label each of the bars: large fish, small fish, invertebrate animals, and finally, algae at the base of the pyramid. Here, you get 1 mark for correctly labeling the x-axis and having a suitable scale, a second mark for labeling each of the bars correctly. 2 marks are also awarded for correctly plotting the width of all the bars. And, as I just mentioned, it has to be within half a small square of the correct value. Calculate the percentage of the biomass lost between the algae and the large fish. Give your answer to 2 significant figures. As a reminder, the biomass of the algae was 840 and the biomass of the large fish was 10 grams per meter squared. To calculate percentage loss, we use the formula initial minus final divided by initial, all multiplied by 100. So let's substitute our values into this. 840, the algae, is the initial biomass, and 10 is the final. This gives us a value of 98.8095. We must then round this to 2 significant figures. Looking to the right hand side of the second digit, we can see an 8, therefore we must round up to 99. This is our answer, 99%. Here, we get 1 mark from the calculation, a second for the unrounded answer, and a third for rounding it to 2 significant figures. Give 1 way that biomass is lost between trophic levels. Well, one of the main ways that this occurs is respiration, in which chemical energy from biomass is converted into heat energy and lost to the environment. You could alternatively have said that it's lost through feces or any excrement. A large amount of untreated sewage entered the river. Many fish died. Untreated sewage contains organic matter and bacteria. Explain why many fish died. This is a 5-mark question, and it requires 5 clear, concise points. So we have our river here, and into it has been dumped sewage. This puts lots of organic matter and bacteria into the river. The bacteria begins to break down this organic material in a process of decomposition. Now, decomposition uses the bacteria to digest the organic matter. The products of this digestion are then used by the bacteria to respire aerobically. Aerobically means it is respiration that requires oxygen. Thus, the concentration of oxygen within the water decreases. Fish need oxygen to respire aerobically, so with less oxygen, less respiration can happen within the fish, thus they have less energy, hence they die. Let's compose our answer. Bacteria decay organic matter by digestion. They respire aerobically, lowering the O2 concentration of the river water. Fish have less oxygen, hence reduced energy supply, so die. It's important to notice that although we have been given loads of space for this answer, 5 marks can be awarded from only a relatively short amount of prose. With biology, clear, concise points is the way to gain the most marks. In this case in particular, the first mark comes from stating the bacteria decay organic matter, the second for stating how, by digestion, the third for discussing their aerobic respiration, the fourth for talking about this lowering the O2 concentration of the river, and finally, a 5th mark for stating the effect on fish, that they have a reduced energy supply. Today we're going to be solving a GCSE Biology question about genetics. Scientists want to breed cows that produce milk with a low concentration of fat. Figure 10 shows information about the milk in one group of cows. The cows were all the same type. Let's have a look at figure 10. Well, we can see it's a bar graph here, and we have number of cows on the left and the percentage of fat in the milk on the x-axis. In figure 10, the mean percentage of fat in the milk is equal to the modal value. Give the mean percentage of fat in the milk of these cows. Well, the modal value is the most frequent value, and in the bar graph, it will be the one with the highest peak, the highest bar. So looking back at figure 10, we can see this is here, which is the mode, and it was also the mean, so just the highest bar. This is equivalent to a percentage fat of 3.7%. Thus, this is our answer, 3.7%. A student suggested the percentage of fat in milk is controlled by one dominant allele and one recessive allele. How many different phenotypes will this produce? Tick one box. Well, to answer this, let's first think about the possible genotypes that will result. We'll call the dominant allele here F with a capital F to represent fat, and the recessive allele F with a little f. So the possible genotypes are homozygous dominant, heterozygous, and homozygous recessive. In this case, you get the same phenotype for the first two, since you need only one dominant allele to be present to show the dominant phenotype. The third, homozygous recessive, however, would show a different phenotype. Thus, there are two different possible phenotypes. Give the evidence from figure 10, which shows that the percentage of fat in the milk is controlled by several genes. Now, if a phenotype is controlled by several genes, then we expect a larger range of phenotypes. If it was controlled by a single gene, we would expect to see only two different possible phenotypes, as we discussed in the previous question. So a bar graph would look like this. However, with many different genes controlling a given phenotype, you'd have a more spread out graph that looks a bit like the normal distribution, that is a bell-shaped curve. Looking back at the figure, we can see it agrees with the latter description, in which there is a large range of phenotypes. So this is our answer. There is a large range of phenotypes, or a large range of values for the percentage of fat in the milk. Or you could alternatively have said that there's not only two values for the fat. Or finally, you could also have said that there's in-between values, as in there's not only two extremes. One of the genes codes for an enzyme used in fat metabolism. In this context, the metabolism of fat means the synthesis of fat, the production of fat in the mammary glands of the cow, that is, its breast tissue. A mutation in this gene causes a reduction in milk fat. The mutation changes one amino acid in the enzyme molecule. Explain how a change in one amino acid in an enzyme molecule could stop the enzyme working. So to explain, we need to describe what is happening and then how this is actually going to affect the biological process. Well, let's think about this. Well, normally, an enzyme will have a specific active site. But following mutation, this active site may change shape, so it is no longer complementary to a given substrate. This is the way in which an enzyme would become non-functional. So the active site is no longer complementary to the substrate. But how would this actually happen? Well, remember that amino acids are the building blocks of proteins. So a change to a specific amino acid could change the type of protein produced by changing its specific 3D structure. So let's write our answer. A different protein is produced with an altered active site, as we already discussed. The substrate does not bind, so enzyme substrate complexes cannot form. Enzyme substrate complexes are what form to catalyze the reaction of the substrate. So let's have a look where marks are awarded in this question. The first mark comes from stating a different protein is produced, the second from discussing the change to the active site, and finally from the result of this, that it will no longer be complementary to the substrate, hence no enzyme substrate will form. The actual terminology of saying enzyme substrate is not necessary for this question. The scientists found one cow with a mutation. The cow's milk contained only 2.9% fat. Figure 11 shows the percentages of fat in the milk of cattle related to the cow with the mutation. The values for male cattle are the mean values of their female offspring. This is because males do not directly produce milk. Let's have a look at the key to this diagram. Black circles are females with low-fat milk. Black squares are males whose female offspring have low-fat milk. White circles are females with high-fat milk. And finally, white squares are males whose female offspring have high-fat milk. Looking at the genetic pedigree, we can see that all of the individuals, 1 to 6, have high-fat milk, since it's prior to the mutation occurring in the lineage. We then see that individual 7 is the cow in which mutation occurs, which has low-fat milk. This is a female, and it mates with a male whose offspring produce generally high-fat milk. They produce a range of offspring, from 9 to 19, some of which have high-fat milk, or have offspring of high-fat milk, and others which directly, or the offspring of which, produce low-fat milk. Well, the question asks us the following. It states that animal 8 is homozygous. The mutation in animal 7 produced a dominant allele for making low-fat milk. Give evidence from figure 11 that animal 7 is heterozygous. Well, let's have a look back to the diagram and discuss this. Well, were the mutation in the cow to be homozygous, then all of its offspring would have low-fat milk, since only one copy of a dominant allele is required in an individual for the phenotype to be expressed. Since some offspring have full fat and some low fat, we can deduce that it's heterozygous. So our reasoning is that it produces some offspring with high fat milk. Animals 7 and 8 produced 11 offspring. These offspring were produced by in vitro fertilization, IVF. These embryos from IVF were transferred into 11 other cows. Suggest where IVF and embryo transfer were used rather than allowing animals 7 and 8 to mate naturally. Well, let's consider that 7 and 8 did mate naturally. Then, each mating season, which is roughly once a year, they could only produce one calf, or perhaps two if they were twins. This is because the female, from which the egg comes, would have to then nurture and gestate the fetus and embryo within her own womb, which can only have one at a time. However, using IVF and injecting embryos into different cows allows several offspring to be produced simultaneously. This is our answer, since it allows more offspring to be produced at the same time. Draw a Punnett square diagram to show a cross between animals 7 and 8. Identify which offspring produce low-fat milk and which offspring produce high-fat milk. Use the following symbols. Capital D equals dominant allele for making low-fat milk, and lowercase d equals recessive allele for making high-fat milk. Well, let's just remind ourselves of animals 7 and 8. Well, 7 is heterozygous, and 8 is homozygous, recessive. Individual 7 was also a female, and individual 8 was a male. It's important to state where each gene comes from when drawing our Punnett square. So that is identifying the male and female. So we have to lay out our Punnett square, as shown, in a 2x2 grid. Make sure you make the grid large enough to state both the genotype and the phenotype. Fill out each of the squares by writing 2 letters in each, from the female and male allele which corresponds to the given square. So for example, the top left square has a capital D, a dominant allele from the female, the mother, and a lowercase d, a recessive allele from the male, the father. Now we have to identify the phenotypes. Well, anything with at least one dominant allele, that is a capital D, will have a low-fat phenotype. So the top 2, the heterozygous individuals, have low fat, and the bottom 2 have high fat, since they have 2 recessive alleles. They're homozygous recessive, so express the high fat phenotype. There are 4 marks available, so let's look at how they are awarded. You get 1 mark each respectively for correctly identifying the male and female genotypes of the parents, and then you get a further mark for identifying correct genotypes in each of the offspring, and then for identifying the correct phenotypes. The scientists want to produce a type of cattle that makes large volumes of low-fat milk. The scientists will selectively breed some of the animals shown in figure 11. Describe how the scientists would do this. Now it's a 4-mark describe question, so you want to make 4 clear points, and we do not need to explain the process. Remember that. So we're trying to selectively breed these individuals. We have to make sure in our answer that we refer to both characteristics. When selectively breeding them, we are breeding them for simultaneously having a large volume and also low-fat milk. In selective breeding, we are choosing the best individuals for a given characteristic, allowing them to breed together. Then from the offspring, we once again choose the best individuals and allow them to breed. This process continues, and almost in an accelerated version of evolution by natural selection, we rapidly produce individuals with the phenotypes that we want. In this case, low fat and high volume. So let's write this down in 4 clear points. Well, first of all, we want to find the female with the highest yield and lowest fat milk. We then also need to find a male whose offspring have the same characteristics. Next, we want to cross these individuals. In this context, cross means allow to breed, to produce offspring. We then want to select the best offspring for both characteristics from each generation, and then here's the key idea, we have to repeat for several generations. Although it's accelerated natural selection, it does take a relatively large number of generations to produce the desired phenotype in the offspring. So let's have a look at where the marks come from. Well, first of all, from stating to find the best female for the given scenario, and the same for male. The next mark comes from crossing the individuals, and the 4th and final mark comes from repeating this over several generations and choosing the best offspring from each. Hi, today we're going to be answering a GCSE Biology question on speciation and taxonomy. Figure 12 shows a ring-tailed lemur. You can identify it by its ring tail, hence the name. Table 5 shows part of the classification of the ring-tailed lemur. We have on the left the classification group and the appropriate name for the lemur. Complete Table 5 to give the names of the missing classification groups. Well, the best way to remember classification groups, I find, is a mnemonic. For example, this one on the left. Delicious king prawn curry or fat greasy sausage. Now in fact, you can ignore the first of these, delicious, which stands for domain, but the rest stand for kingdom, phylum, class, order, family, genus, species. Give the binomial name of the ring-tailed lemur. Use the information from Table 5. Well, binomial means dual, it means 2 parts to the name. For the ring-tailed lemur, and in fact for any species, this is composed of, first of all, the genus, followed by the species. The genus will have a capital letter, whereas the species will have a lowercase letter. This will be underlined if drawn out by hand, or in italics if printed. So Using information from Table 5, we can see the genus is Lemur and the species Cata. Hence the binomial name is Lemur Cata, with a capital L, lowercase c, and all underlined. Now, for this question, there's no need to underline it, as in the mark scheme you get marks without underlining your work. Lemurs are only found on the island of Madagascar. Madagascar is off the coast of Africa. Scientists think that ancestors of modern lemurs evolved in Africa and reached Madagascar about 50 to 60 million years ago. Today, there are many species of lemur living on Madagascar. Figure 13 shows information about water currents, and Figure 14 shows a distribution of 3 species of lemur on Madagascar. Looking at Figure 13, we can see there are several water currents, 50 to 60 million years ago, that are heading towards Madagascar, whereas today, this is not the case. Figure 14 also shows that the 3 different populations are disparate and separated geographically. Note also the scale of these maps. There is around 300 kilometers between Africa and Madagascar, even at its closest point, so it's very unlikely that anything will be able to swim across. We'll bear that in mind for later. And so suggest how ancestors of modern lemurs reached Madagascar. The command suggest means to give an educated guess of what could have or is most likely to have happened. So, what is most likely in this case is that they were carried by favourable currents, which we already identified are heading towards Madagascar from the coast of Africa, 50 to 60 million years ago, on masses of vegetation, as, since we already said, is too far to swim. Describe how the ancestors of modern lemurs may have evolved into the species shown in Figure 14. So this is a 5-mark describe question. So we need to get 5 clear, concise points without explaining anything. So essentially what we're being asked to find here is to describe the process of speciation. How individuals that arrived as lemurs onto Madagascar form into different species over time. Well, how did this happen? Well, different populations on the island, once the lemurs migrated there, became isolated from one another. This was the first stage. There was considerable variation in the habitats of these different populations. This is significant because different habitats will impose different selection pressures that eventually result in the formation of different species. For any form of evolution to occur, we require extensive genetic variation. So this will be our next point. There was substantial genetic variation within lemur populations. This ensures there is enough different alleles which result in advantageous alleles increasing in frequency within a population. Those individuals that are better adapted survive to reproduce. Thus, they pass on the favourable alleles, which are in their own bodies, to their offspring. Over time, as this occurs, the populations will become better and better adapted to the environment in which they're in. And eventually, individuals of one population cannot reproduce, as in cannot produce fertile offspring with individuals from another population. This is the definition of species. Hence, they are separate species. Since for individuals to be of the same species, they must be able to reproduce, to produce fertile offspring. So let's have a quick look at where we get the marks from this question. First of all, from stating the geographical isolation of different populations, or just the isolation is sufficient. Then stating that there was variation, first of all, 1 mark in the habitats, and second of all, another mark for the variation in genetics. The 4th mark comes from essentially a very brief description of natural selection. That is that some individuals are better adapted, and hence their alleles will be passed on to the offspring. And the final mark is for stating that over time, speciation does occur. That is, separate species form, since individuals from one cannot produce fertile offspring with individuals from another. There are the full 5 marks.